Hello, everyone. So welcome to the GIGA session. We have a great array of speakers. I will not be giving much of an introduction. I'm Jean-François Flo, by the way, from Brussels in Belgium. And I will right now introduce the president of the GIGA Association, so the Global Invertebrate Genomics Alliance, Joe Lopez. Joe, if you would like to give your introduction.
Hi again. Good to see everybody. I just wanted to say good morning, and I'm very happy to introduce our GIGA session today. Firstly, I'd like to thank all the Biodiversity Genome 2020 organizers, Mark, Blackster, Harris, Eric, Sophie, Chloe, and all for their enthusiastic and support and assistance. I'd also like to thank our GIGA co-chairs who assisted in contacting and arranging our speakers today. And we also tried to get them listed. Jean-François Flot is actually a co-chair, as well as Bharat Patnak. We've also had 13 great posters at this first online meeting, Biodiversity Genomics, which I hope folks had a chance to visit. I don't want to take too much time right now because we want to start our 7 speakers talks, but in reality, there's a lot of progress going on in our community in the invertebrate genomics field, and I think you'll see part of that today. You may already know the brief history of our alliance, which began only 7 years ago as an idea and an offshoot of the Genome's 10,000 initiative. For more background, I point you to our refurbished GIGA website and 2 white papers that are there to explain our overall mission. Before I get to our big announcement, which we've been advertising a bit, I'd like to highlight a few recent milestones. Firstly, GIGA became a nonprofit community this year, and will continue to embrace a bottom-up grassroots philosophy, applying as many ideas research as possible, driven by the membership, which is now about 200 strong. We welcome all and any, not just genomicists, but a wide variety of biologists to contribute to genomics progress. The membership link and support is on the community webpage. Secondly, the Gordon and Betty Moore Foundation, along with the Wellcome Sanger Institute, has recently provided significant funding for a phase 1 project to sequence 200 aquatic symbiotic genomes via 4 taxon-specific hubs. GIGA will be leading a hub to coordinate photosymbiosis invertebrate sequencing, which includes corals, mollusks, sponges, tunicates, aseals, and other invertebrates. We joined 3 other hubs focused on sponges, corals, and protists. They are now calling for a phase 2 proposal call for 6 more hubs. These initiatives all help to build a wider genomics community, as GIGA is also part of the Earth Biogenome Project. And lastly, for our advertised big announcement, which is consistent with GIGA's mission to support genome education and early career research, we are now offering a competitive student fellowship of $2,000, which can be applied towards any research on invertebrate genomes. Genomic projects. The GIGA Fellowship in Invertebrate Genomics is open to any meritorious graduate student from around the world involved in any area of invertebrate genome research, except insects and nematodes, encompassing wet lab, computational, and theory. The funds cannot be used for travel or collection of animals. We will be accepting applications immediately. You can write Dr. Heather Bracken-Grissom before the deadline of November 13th, 2020. More details are on the GIGA website, giga-cos.org. With that, we look forward to this session on fascinating animal invertebrate genomes. And I turn this over now to Dr. Jean-François Flot.
Well, thanks a lot, Joe, for this very great announcement. And now we have, as I mentioned, a series of speakers. And the first speaker will be Gerd Verheide from Munich in Germany, who will be telling us about the Ignite ITN initiative that emanated in part from GIGA.
Hello, everyone. Welcome to my presentation on the European Training Network Comparative Genomics of Non-Model Invertebrates, ITN Ignite. My name is Gerd Verheide. I'm the initiator and coordinator of this wonderful program, and I'm presenting this here on behalf of the whole Ignite consortium, which I will introduce to you in a minute. So European Training Network, what is an ITN? What is such an endeavor? I just want to start off with giving you a small, short introduction into what ITNs are. ITNs are called is a measure as an action from the Marie Curie actions under the Horizon 2020 program of the European Union. And common features of such innovative training networks are that they are an international network of organizations. So some scientists and organizations get together to develop a joint research training or doctoral program. So you see already here, the focus is on joint research training. And these programs are meant for so-called early stage researchers, ESRs. Those are researchers with less than 4 years of research experience and those who do not have a PhD yet. So they get trained to achieve or receive a PhD after their tenure in this program. One of the important parts and components of such an innovative training network is mobility across countries. So you will not be eligible to take up a position in the country where you have resided for a specific time prior to appointment. So definitely not in your home country. So the European Union really wants to get people moved across countries. And eligibility is not restricted to Europeans, so you can come from all over the world to participate in such an ITN. The ESRs receive in these programs multidisciplinary training, mainly through individual research projects, but also through joint research and joint training activities. They receive high quality supervision and career guidance. And another important component of an ITN is that they receive meaningful exposure to the non-academic sector, industry, NGOs, museums, collections, etc. And they also receive training in transferable skills, entrepreneurship, management, and communication, because the graduates from such a program should not only be employable in the academic sector, but they should go out in various fields, including the industry. And another important component of such a training network are the network-wide training events and activities where exchange of knowledge happens within the network. And I come back to that in a minute. So this is the background and the framework. So what is the ITN Ignite? It was born out of the second GIGA workshop that we organized in Munich 2015. And I'm very grateful that I have the pleasure and the opportunity to present the Ignite training network here in this GIGA session. During that workshop in 2015, we had breakout groups and we're talking about funding and training, and we identified training is one of the most important aspects of GIGA. And I said, okay, let's try the ITN scheme of the European Union. They're highly competitive. There's about a 6 to 7% chance of getting such a network. And luckily, we were lucky to get one with a total budget of 3.8 million euros for 48 months. That is distributed. The money is distributed in this case here, to 14 beneficiaries, and the program runs from 2018 to 2021. We were lucky to be able to recruit 15 early stage researchers for 36 months to carry out research and training within the network. And this is a fantastic group of people. You see some heads here, some names and heads here. They are distributed all over Europe, from Bulgaria to Portugal to Ireland and Norway, and some in Central Europe. And some of them, like Ferenc and Anne and Nadej and maybe someone else, will present their work from the program here at this Biodiversity Genomics Conference. We also have a few partners. The Queensland Museum is a museum in Australia. Alpha Biotoxin is a company in Belgium. The Leibniz Rechenzentrum north of Munich provides high computing infrastructure and power, and the Institut Pasteur and INRIA are also partners who are mainly focused on genomics and molecular biology. So what are the research goals of Ignite? First of all, we want to improve the sampling and analysis of invertebrate genomes. On the left-hand side, you see a tree with this 30-odd phyla of animals. Many of them, most of them, are in fact invertebrates. And the blue bars represent species described. The red bars represent the number of species with sequenced genomes. And you see a few blanks here where we do not have genomes from several lineages, and we are set out to especially enhance genome sampling from undersampled branches of the animal tree of life. And the green stars here represent the lineages where we are going to obtain data from, and the number is the number of the ESR. Along with sequencing genomes and analyzing genomes, we are going to extend the toolbox for the analysis, and that includes the development and deployment of innovative production level software. And we have several of our ESRs involved in these such endeavors. IGNITE has the following core training objectives. We convey broad interdisciplinary knowledge on organismal biology, on physiology, on evolution, on biogeography, development, etc. We provide technical laboratory skills required for genomics and computing skills, technical computing skills in programming and bioinformatics to analyze genomes. So the ESRs, independent of where they come from, whether they come from organismal biology already or from computer science, they get a holistic training in all aspects of animal biology and genomics. They get trained in effective dissemination and communication of results to the scientific audience and also to the public. They get trained, we provide relevant academic and non-academic transferable skills, grant writing, intellectual property management, Nagoya Protocol, Convention of Biological Diversity, and so on. And another very important aspect is that the ESRs get trained in establishing a strong, long-lasting European network of invertebrate genomics together among themselves and also together with the PIs. I mentioned earlier that network-wide training events are a very important and interesting endeavor in such a ITN. And the first one we had in Split in Croatia in summer 2018. Beautiful place, the Mediterranean Institute for Life Sciences. I can highly recommend this place if you want to have small meetings. It's right at the edge of the Adriatic Sea. Outside of Split, and there we took the students, the ESRs, really down to the bottom, so to speak, because we took them out on the boat. You see on the left top side, we were explaining the habitats. We took them snorkeling. Some of them have never had the opportunity to snorkel and see invertebrates alive underwater, which was really cool. We sampled the plankton critters. We took the samples to the lab. We database them, introduce them to voucher specimens and morphological descriptions. And then we train them in the first steps of genomics, like DNA extraction, preservation, etc. And at night we had lectures on the general biology of the several invertebrate lineages. This is Andy Heinol here talking about molluscan larvae. During that very nice 14-day network training event, the students really bonded and really got together and formed a really nice coherent group. And they identified that some had strength in this area, some had strengths in that area, and they wanted to get together and teach. And they approached me whether they can have their own workshop. And yes, they did have their own student programming workshop in 2018, again in Croatia and in Split at the Mediterranean Institute of Life Science, without any supervisors, where they were just self-organized, their teaching, and they taught each other what was their background that was highly successful and very much appreciated. We had two more training events in 2019 in Bagnols-sur-Mer in France and Zagreb, which were also highly successful. Unfortunately, due to the Corona situation, this year everything came to a grinding halt, and we hope to be able to continue that string obviously next year. So the remaining few slides, I would like to introduce to you a few of the projects. We're focused on the training now. Just a few snapshots and highlights of the research our ESRs are doing. And I start off with an example from one of my ESRs, the one I have the pleasure to supervise, Xenia Juravel, and she is focusing on deep metazone phylogeny. As some of you may know, that some of the relationships in animals are highly disputed, especially those close to the root of the animal tree of life. The relationships of the non-bilaterian phyla are highly disputed. And Xenia is taking a different approach away from analyzing primary sequence data in terms of long phylogenomic amino acid alignments. She's focusing on analyzing the genome content of genomes. And to cut the long story short, here's one of the intermediate results, and her results are consistent with the porifera sister hypothesis, that sponges are the sister to the remaining animals, but also consistent with the hypothesis that the Sena solomorpha are sister to the rest of the bilateria. It's still ongoing, and I'm looking forward to seeing the final results. Kenneth is an ESR at the National University of Galway in Ireland, and he is working with Grace McCormick, and they're interested in natural products chemistry, so they're sequencing a sponge with long read and short read technology, and they have already identified a gene cluster that may be responsible for the production of bioactive compounds. And they are now going more into depth to analyze this cluster. And Jimin Chen is analyzing sequencing de novo genome of a slug. Her project was really impacted by COVID because she wanted to go to the Northern Atlantic to sample some pelagic mollusks. And so that wasn't possible. So she focused now on a snail that we have here in Bavaria. She's also based in Munich at the Zoological State Collection. And she's also using long read and short read technologies, nanopore in this case, and Hi-C to sequence and assemble this large genome, relatively large, 1.5 gigabase large genome, but was very good scaffold N50 of 64 megabases. And finally, ESR 15, Maria at Pensoft is doing a very interesting project because she's interested in semantic publication, dissemination, visualization, and reuse of biodiversity-related genomic and metagenomic data. And what she aims to do is to make biodiversity and genomics data more fair, findable, accessible, interoperable, and reusable, so that different platforms and different databases can automatically harvest the data that is generated. And with this, I'm going to stop. I thank you for your attention. I thank the fantastic network. You see all the names here, and the supervisors are in italics. It's a great group, and I think we're making very good progress in training and in research in invertebrate genomics. And I would like to thank the European Union for providing funding for this fantastic network. Thank you very much.
This was a very interesting presentation from Gert, who unfortunately is not here today to answer the questions, but we have here Mika Aitan with the project manager, and we will be answering the questions asked. So I see your first question online. What is the relationship between GIGA and I5K? Miki, what could you say?
Hi. First of all, apologies from my boss. He couldn't make it. He's up in the mountain, so there's no internet connection. So I'm trying to answer all the questions. So as said, I'm project manager, and I'm trying to keep this all together, this endeavor for the next 1.5 years to go. So there is quite some overlap between GIGA and I5K in that respect, that some people that are in this consortium also are working in the I5K. Apart from that, we don't aim to sequence any insect species that are not any insect species and nematodes. So there's only overlap in terms of people that working on one or the other or both.
Yeah, Thank you. This brings me to another question I wanted to ask you. What do you think were the main challenges you faced during the managing of such project? What advice could you give to such a group who would like to make up such a project together?
Well, first of all, the very important thing is to find reasonably good or excellent students. So that was quite challenging in the beginning to find people from all over the world. And we made it open all over the world because we wanted to have the best people to come to Europe and to get the best training. And so that was quite an endeavor in the beginning. But we managed after a couple of months. Some projects started a bit later because it was difficult to find some. But that was the most important thing. And then setting up workshops, that was also quite time consuming but very interesting because of all the different topics that we are going to cover in this only 4 years. So that lots of teaching. So not only time to spend in a lab, but also sitting in the classroom and getting taught state-of-the-art genomics.
Very true. Maybe time for one last question. How much has the network activity been impacted by COVID-19? I mean, for such an international venture, sounds like tricky given the restriction movement, for instance.
Yeah, so we tried to have our last workshop to be held in Porto in April. Of course, it was not able to do that. So we are still in the phase of trying to, when the cat is helping. So I'm in the natural habitat, which is our kitchen. So we're trying to bring this online in a bunch of blog sessions to make it happen still. So it was quite tricky in the beginning, but we're now getting close to the point where we can have all these online sessions to be run by the end of this year. So we were affected also in terms of sampling for the sequencing. So a bunch of projects had to change their direction because there was no way to get in the samples. So more and more towards bioinformatics instead of sampling in the beginning.
Thank you very much. I think it's time to move to our next speaker in line, so Kate Castellano, and I look forward to this great talk. Thank you.
I'd like to start by thanking the organizers for giving me the opportunity to speak today. I'm really excited to share my thesis work so far, which focuses on the genome dynamics of Salpa thompsoni, which you can see pictured here. And while at first glance Salpa look like jellyfish, they actually are not. They are tunicates found in the phylum Chordata, and they're one of the most closely related invertebrate species to humans. And while salps are found in all of the world's oceans, my research focuses specifically on Salpa thompsoni because it inhabits the Antarctic Ocean. And as we all know, the Antarctic is experiencing large-scale environmental changes, including sea ocean temperature increases as well as sea ice melt. And accompanying these changes are large-scale population density increases in Salpa thompsoni, particularly in the Western Antarctic Peninsula. And while this is good for salp, we find that with these population increases, we're seeing over twofold decreases in other important zooplankton species that live there, such as krill, which serve as the basis of the Antarctic food web. Therefore, these large-scale population increases can have huge effects on the Antarctic food web and are having large effects on the Antarctic food web. However, there's still a lot of information lacking about salp and how they're able to perform so well in a rapidly changing environment. However, one of the reasons they're able to do so well has to do with their reproductive mechanism, which alternates seasonally between solitary individuals, which asexually produce chains or aggregate salp, which look like this. And these chains will sexually reproduce with each other to produce new solitary individuals. And these new solitaries will then, in the fall and winter, descend down deep into the water column where they'll spend the winter before coming up and starting this process all over again. And it's during this spring and summer seasons where we're seeing these huge population density increases or blooms, where there's extremely successful asexual reproduction to the point where there are so many chains that they completely take over, like they're taking over your screen right now. And by doing so, they outcompete these other organisms for food and other resources. However, since we've seen these population increases since about the 1950s, we really lack a genetic and genomic understanding of this reproductive mechanism. So my thesis focuses on trying to understand the genomic mechanisms supporting successful population blooms of Salpa thompsoni in a rapidly changing environment. And to do so, the first thing I needed was a genome assembly. And when I first came into the lab, we did have a version 1 draft, which was done with short read sequencing. However, this assembly only covered about 50% of the expected genome size, which is 600 megabases. Additionally, it was extremely fragmented with over 180,000 contigs with an N50 of 1.5 kb. Additionally, when I tried to do another short read sequencing experiment with linked reads, I found that my assembly was also extremely fragmented and had a really small contig N50. And if we look closer at this version 1 draft, we find that the repeat content is 33%. So this explains why we consistently get fragmented assemblies when we use short read technology. So for version 2 of this genome, I used only long read sequencing, particularly Oxford nanopore. And I've done about 10 iterations of this assembly. Here I'm showing you just the final pipeline. And I don't have time to go into the details, but I'd be happy to talk more about this during the questions. And I found that long read sequencing greatly improved this genome by covering the expected genome size, as well as greatly reducing the number of contigs from 180,000 to 8,000, and also increasing my contig N50 from 1 kb to 191. Additionally, when I do a preliminary analysis of the repeats, I find that it's 67% repetitive, which is on par with what we expect based on our version 1 draft assembly. Unfortunately, about 41% of these repeats are unclassified, so I'm currently working on a modified repeat annotation pipeline to better classify these repeats to better understand the repeat landscape of this genome. And while the long read assembly improved contiguity of the genome, I also found that it improved completeness when we analyzed this with BOOSCO. And I found that I increased my completeness boost scores from about 40% to 77%. I reduced my fragmented boost scores from 27 to 6%, as well as reduced my missing boost scores from 30 to 16%. So overall, long read sequencing greatly improved this genome. However, it is still slightly fragmented, and this probably has to do with the high repeat content. And while assembling a genome with high repeat content is difficult, I also ran into other problems while I was trying to create this genome. And that was at the sequencing level. So when you do Oxford nanopore sequencing, typically you get these really nice graphs with the lime green bar representing the number of active nanopores that are actively sequencing your DNA. And you should have a really small dark green bar representing your active pores that are not sequencing. However, with SALP, I always get something that looks like this, where I have really low sequencing efficiency and never more than 50% of my pores sequencing at a time. And I worked really closely with Nanopore trying to troubleshoot why this might be. Typically, when you see something like this, it means there's a library concentration issue or a contamination issue. However, neither of these are my problem. And the other thing it could be is a ligation efficiency issue. And this actually led me to look more into secondary structures with the hypothesis that there could be a secondary structure inhibiting ligation efficiency and therefore reducing the number of available DNA to be sequenced through my pore. And one of these secondary structures I looked into are called G quadruplexes. And these form in the genome when you have stretches of guanines and they base pair with each other, forming a quartet. And so when you have really long stretches and multiple quartets, they can fold on each other, forming these really stable structures called G quadruplexes or G4s. And these are found within the genome, typically around your telomeres. They've also been shown to be important for key genomic functions such as transcription and replication, epigenetic regulation, genome stability, and DNA elimination. And so based on the ability to look for these DNA sequences in the genome and the high repeat content of the SALP genome, I decided to see if this could be a possibility. And I was extremely surprised to find that 91% of my contigs contain G4 sequences. And here I'm showing you contigs on the x-axis and the number of G4 sequences found within each contig on the y-axis. And so as you can see, these G4 sequences are found abundantly throughout the genome as well as within many of the contigs. So why would this be? What is their function and why are they so abundant in this genome? And is this something that we see in all tunicates? And when I compare them, I find that the answer is no. So here what I'm showing you with the purple graphs are the number of contigs in each of these tunicate assemblies. And the black dots here, I'm showing you the percent of contigs that contain G4 sequences. And what you can see that both Salpa thompsoni and Salpa aspera have a much higher abundance of G4s within their genomes compared to these other tunicate species. And so this suggests to us that the abundance of G quadruplexes in these genomes is specific to the Salpa genus. But why would that be? And what is different about Salp versus other tunicates? And one of the differences is their alternating reproductive life history, which I mentioned earlier. And G4 sequences have been implicated in DNA elimination. And so maybe they could be acting in this way. The other difference is that Salps tend to inhabit deep sea locations. And G quadruplexes have also been implicated in genome stability, so potentially they're playing a role here. And when I compare my Salp genomes with other organisms that we know go through DNA elimination, we find that the abundance of these G4 sequences is similar to what we see in the Salp genome, particularly the lamprey, which eliminates DNA between their somatic and germline tissue. Additionally, if we compare to a deep sea genome, the deep sea snailfish, we find the abundance of G4s in this genome are also similar to what we find in the Salp genome. And here you can also appreciate, if you compare the deep sea snailfish to the shallow snailfish, that there's a large difference in abundance of these G4 sequences. So this suggested to us that the role of G quadruplexes may be multifold. It may be important for both genome integrity as well as DNA elimination. And so I'm currently following up both of these hypotheses, but particularly for the DNA elimination question, I have created a developmental transcriptome, and it encompasses both sexual and asexual stages, including multiple solitary adults as well as their asexually produced embryos, and multiple aggregate adult stages as well as their associated embryos. And I've completed this transcriptome and it has a BUSCO completeness of 97%. Additionally, when I look at differential expression of genes between these different stages in the reproductive cycle, I find gene clusters that are differentially expressed together between the different embryonic stages, so from aggregate to solitary, as well as clusters of genes that are differentially expressed between the solitary adults and the aggregate adults. And I'm currently working on identifying exactly what genes fall into these clusters and their role in this reproductive life history. So in conclusion, what I've told you today is that I've created a more comprehensive and complete salpintomycin genome. And by doing so, I've been able to validate the high repeat content, and I've identified an enrichment of G quadruplex sequences in these genomes. Additionally, I have created a developmental transcriptome and identified co-expressed gene clusters between the different developmental stages. And currently in the future, what I'm trying to do is look at gene and repeat associations with my G quadruplexes to better understand what their role may be in the salpintomycin genome and how they may be supporting this complicated reproductive life history. And with that, I just want to say a quick thank you to my mentor and PI, Rachel O'Neill, for all of her support through this project, as well as my amazing lab members. And also Dr. Ann Buckland and Dr. Paula Battaglia for the SALP samples and all of their SALP knowledge, and Dr. Jill Wegerson for all of her genome and transcriptome help, as well as our amazing sequencing and computational biology core centers, and my funding. And with that, I'd be happy to take questions.
Thanks a lot, Kate, for this great talk. I have a bunch of questions. I hope we can answer them all. One first question is, how did you estimate your genome size? Was it just from the assembly, from the data themselves, or did you have an independent estimate?
Yeah, with the original assembly, we did an independent genome size estimate using qPCR.
Okay. Wondering also, what Busco database did you use precisely?
Yep, I used the Metazoa, both the version 9 and version 10.
And did you use a -- long option or the regular default way of running Busco?
Yeah, I used both.
Okay.
Yes.
Do you have any idea how abundant the G quadruplexes generally are in other taxa, except the one you mentioned?
Oh, yeah. So the abundance of G quadruplex varies. There have been quite a few studies done in bacteria that have shown certain taxa have a higher abundance than others, and it seems to be associated with, adaptation to extreme environments like heat and things like that, or they see higher abundances in things like tetrahymena that are shown to perform DNA elimination. So it seems to vary, and it seems to, I noticed that it varies considerably within a genome as well. And that's why I think it's really important for my next steps to look at the locations in the genome, where they are, and how what that means about their function.
Thanks. Someone asked, how did you assemble the transcriptome using what software?
Yes, I used Trinity to assemble the transcriptome. Mm-hmm.
Okay. Another question, why would the G4 structure inhibit the adapter ligation efficiency? Do you think it may instead rather block the pores?
Yeah, so originally we thought one of the problems was a ligation efficiency where I was loading enough DNA, but it didn't have the appropriate adapters to be sequenced through the pores. And so we thought maybe that was a secondary structure, and I had looked into multiple secondary structures, and G quadruplexes were just one of them that I decided to look into, and I actually didn't expect to find anything interesting. And so I was surprised by that. We didn't know what structure would be there, or if in some way it was folding so that the ends were unavailable for ligation. And so I don't know about G4s in particular, but that could be the problem.
And wondering whether did you look at these G4 sequences in Sionna, for instance, since they're closely related to thalassians?
In Sionna? Yes. So I did actually, and they actually do have a high abundance of G quadruplex sequences as well. And what I didn't talk about in the cell reproductive cycle, I just talked about asexual and sexual reproduction, but the chains, the sexual chains actually are also hermaphroditic. So they start out as female and then switch to being male at some point. And that's something that also happens in Siona, they're hermaphroditic. So that also supports the idea that the G4s are somehow involved in regulating this reproductive switch.
And one last question, that one from me. You mentioned 10 iterations of your pipeline,
Yeah.
but what are the part that get iterated actually?
Yeah, so some of the iterations were the amount of data I was using as well as the type of assemblers. So I tried multiple different long read assemblers that use different methods for assembling because I wasn't sure with the repeats what would be the best. As well as polishing, I tried I tried nanopolish as well as Medaka to identify which one would be better for this genome. And now I'm working on the repeat and gene annotations, trying to get those right.
Thank you very much. We're going to move to a completely different taxonomical group, sponges, with Cecilia Konako's speech.
I'm Cecilia Konako from the Marine Science Institute of the University of the Philippines. The title of my talk is Adaptive Capacity of Sponges in a Changing Ocean, Insights from Demosponge and Calcarean Transcriptomes. The work that I will show you today was conducted mostly by my student, Nino Dan Busadas. So sponges, or poriferans, are the earliest animals. They diverged from other metazoans some 500 to 600 million years ago. Sponges come in all shapes and colors, and they can be grouped into 4 major classes, shown here, which are distinguishable from each other based on their spicule type and morphology. Most sponges have spicules made of siliceous material or organic fibers, except for the calcareans, which have spicules made of calcium carbonate. Despite their seemingly simple form, sponges actually are quite adaptable and can be found in a wide range of habitats. The demosponges, which are the most species-rich group, and the calcareans are quite common on tropical coral reefs. So sponges play a major role in coral reef ecosystems. So here's a sponge. Together with their symbiotic microorganisms, sponges are involved in the cycling of nutrients. They also provide food and habitat for other marine organisms, and some are important for reef consolidation or bioerosion. Previous studies have shown that sponges are also resilient to all types of stressors, such as ocean warming and acidification, which are associated with climate change. And so sponges are predicted to eventually become one of the most dominant taxa on reefs of the future. However, we still understand very little about how different types or classes of sponges would be responding to these future ocean conditions. And so in this study, we wanted to compare the responses of 2 species, the demosponge Neopetrosia compacta and the calcareous sponge Lucetta chagosensis, to varying levels of acidification and warming. We tested 5 different treatment combinations, which are shown up here in the colors. So dark blue represents present day conditions. Light blue is acidification only. Yellow is warming only. And orange represents a combination of warming and acidification based on the RCP6 model. And red represents another more intense combination based on the RCP 8.5 model. After 5 days of sustained exposure to these treatment conditions, we observed that the Neopetrosia fragments remained healthy all throughout. In contrast, Lucetta began to show signs of some tissue necrosis in the warming treatment as well as in the combination treatments. These results tell us that the calcareous sponge is more sensitive to these particular stressors. So now we wanted to understand the molecular events that accompany these observations. And so we performed transcriptome sequencing and differential gene expression. These results reveal that the demo sponge, Neopetrosia compacta, had very few differentially expressed genes. In fact, it only had 89, in contrast to Lucetta chagosensis, which had more than 2,000 differentially expressed genes. The genes could be grouped into 5 main expression patterns, labeled 1 to 4 in this figure. And as expected, the most intense treatments resulted in the greatest number of differentially expressed genes. So for example, most genes showed a downregulation in expression with intensifying treatment, as exemplified by cluster 1, and others showed an upregulation in expression, as exemplified by cluster 5, so with increasing stress. We then wanted to determine the cellular functions that were represented within these different clusters. And to do this, we performed gene ontology analysis. We focused on the genes that were in Lucetta chagosensis because it had more differentially expressed genes. Here you can see a figure showing cellular functions that are enriched within each of the different clusters shown up here above each column. The shading within the boxes represent the degree of enrichment for each of these functions. So for cluster 1, which are the genes that are most expressed in control conditions, we found an enrichment for general cellular functions such as autophagy, metabolism, translation, immune response, gene expression, morphogenesis, and so on. Examples of genes within this cluster include the extracellular matrix proteins, collagen alpha, and hemagglutinin aggregation factor, which may play a role in maintaining sponge tissue integrity. For clusters 2 and 3, we did not find too many enriched functions, but those included signal transduction, extracellular matrix cell signaling, and tissue regeneration. For clusters 4 and 5, we found an enrichment of functions mostly related to protection and defense. The functions listed here include defense response, regulation of protein stability, or protein folding down here, regulation of apoptotic process, cell redox, homeostasis, DNA integration, response to pH, response to bacterium, and cell death from oxidative stress. Examples of genes included within these clusters are heat shock proteins, caspases, and sequestosome. These results tell us that under RCP6 and RCP 8.5 treatments, which are the most intense treatments we use, the sponge has downregulated much of its normal cellular functions and has devoted much of its cellular resources to repairing damage and restoring cellular homeostasis. Now, although the demosponge Neopetrosia compacta did not reveal many differentially expressed genes, we were able to identify within its transcriptome homologs for the differentially expressed genes in Lucetta. And interestingly, when we plotted the abundance of these genes, we found that many of them were actually more abundantly expressed in Neopetrosia under controlled conditions. So those are the genes that are represented by points within this blue shaded region, and we can call those genes front-loaded genes. It is possible that these abundantly expressed stress-related genes contribute to the overall tolerance of Neopetrosia, because as you can imagine, the availability of these transcripts and their protein products would allow the sponge to mount a rapid response to any environmental perturbation without having to transcribe new genes. This mechanism of front-loading has previously been described for corals. So having identified stress-responsive genes in the calcareous sponge Lucetta and their homologs in the demosponge Neopetrosia, we then sought to identify homologs of these genes in other sponge species down here, which have available genomes and transcriptomes. We focused mainly on genes involved in adhesion, receptor activity, apoptosis, and defense. What we found here is that there are species-specific differences in the abundance of certain gene families, but for members of the same class, they typically share similar abundance patterns. So, for example, genes with calcium-binding EGF domains or with NACHT domains and lectin C-type domains are typically more abundant in the calcareous sponges, while genes with scavenger receptor domains, which are these ones, and death domains are typically more abundant in the deep sponges. So this suggests a basis to predict species-specific differences in the stress tolerance and adaptive capacity of sponges. However, it is also important for us to understand how the different sponge species may express or deploy their own stress response toolkit under different conditions. So to summarize, in this study, we've shown that the demosponge Neopetrosia compacta and the calcareous sponge Leucetta chagosensis have very different responses or susceptibilities to varying levels of acidification and warming. Neopetrosia compacta maintains an overall stable gene expression profile, which indicates maintenance of normal cellular functions. Eventually, the sponge may be able to acclimatize or adapt to these new conditions. On the other hand, Leucetta chagosensis revealed large-scale changes in gene expression, indicating disruption of normal cellular functions. Eventually, this may result in disease or mortality of the sponge. And to conclude, differences in the abundance and expression of stress-related genes may contribute to differences in the capacity of certain sponge species to tolerate or adapt to rapidly changing ocean conditions. These species-specific differences in susceptibility to stressors will eventually impact population structure and overall ecosystem function. And so this highlights the importance of conducting further studies to elucidate the responses of more diverse sponge species. So I would like to acknowledge the following people: Michael Nada, Jake Bakiran, Dr. Patrick Kapaitan, and staff of the Bolinao Marine Laboratory for their assistance with this study. Also, Dr. Michelle Kelly, who helped us identify our sponge species. And funding for this work was provided by the Department of Science and Technology of the Philippines. That concludes my presentation. Thank you all for watching.
Thank you, Cecilia, for this great talk. I have some questions for you. So what was the control you used in your differential expression analysis? And what did you compare against, actually?
Thanks for that. Yeah, so we had a present day control. So just if you're interested to know the parameters, it's 28 degrees for us and a pH of about 8. Okay, I was more thinking the question was about internal control, like genes that you were normalizing to. Oh, yes, sorry. For the differential expression, we used, if I remember correctly, EDGE-R. So it's global normalization. We did not compare against any internal housekeeping genes for that particular analysis.
Thanks. And also, how did you annotate your genes with Go terms? Did you annotate impacts? What you find you perform did the way you annotate impact the way what you found in the enrichment analysis?
Yes, it's always one of the biggest challenges, right, with these organisms. We annotated, we collected gene ontology annotation through looking at their best hits in BLAST to other organisms, to the available amphimedon genome. But essentially, we still get quite a bit of unannotated, and I think that really does limit what we can say about the data, since there's so many things that might be interesting that we just don't know what they're about. But then we do other additional analysis. For instance, we also look at protein PFAM domains, for example. In case that there's no matching gene ontology annotation, we could still sort of make a guess as to potential functions of these encoded proteins.
And I'm wondering, because we do a bit of sponge genome sequencing here in Brussels, and it's quite challenging. At the level of RNA-seq analysis, is there also some specific challenges when you deal with sponges?
Sponges are they're quite tough to work with sometimes. There's because they do also have quite a bit of microbial symbionts in there. So that's really something we have to watch out for when we're doing these types of analysis. For transcriptomes, we do mRNA enrichment to try to get rid of some of that, but we still do get quite a bit of microbial sequences. And some sponge species, like I think Neopetrosia, was pretty, mucousy. So it also is a big challenge in terms of RNA extraction.
Yeah, there was one question that came right after, precisely, how did you deprive bacterial and symbiont hits from transcriptome assembly? So if you're enriched for poly A, bacteria should be fine, but eukaryotic symbionts, how did you deal with them?
Yeah. Well, they're still there, whatever they are. So those eukaryotic symbionts are still there. We essentially just try to take everything all together and see what's happening in the entire holobiont, because at this point there's no way that we can separate some of these other associated organisms, except if we are able to compare it to a database and available genome of something else. So that's basically how we do it. We deal with the whole thing as a holobiont altogether.
Well, thank you very much, Cecilia. I think now it's time to move to the next speaker, Ruth da Fonseca.
Hello, everyone. So I'm Ruth Fonseca. I'm an associate professor at the University of Copenhagen. I'll tell you a little bit about our giant squid genome project that just got published this year. So why sequencing the giant squid? Usually I have to answer that question. It is rarely seen, usually found dead, rotting, right? Because it lives in the depths of the ocean and it only comes to the surface once it's dead. We can't really study these squids in the lab. You cannot really make cell cultures of giant squid. I tried asking, no karyotyping possible. The DNA is usually low quality because it is usually found dead, and it's quite heterozygous as other wild animals. So lots of difficulties in the assembly because of that. But I don't have to tell you how often invertebrates are neglected. So it's another invertebrate genome that has been done. So that's a contribution. And in general, we just thought giant squids were really cool. I was working at the time when I started this project with Tom Gilbert, as you can see here, and a truly enthusiast of giant squids. He made this suit himself. And here is Inga that did all the lab work in this project, and she's also an aficionado. So we just thought it was really cool. It was also first described by Dane, so it was Japetus Steenstrup that first connected the legends of this kraken, the hugest monster in the sea that swallows men and ship, and said, well, I think this is actually a giant squid. So that helped with the funding. And when our genome paper came out, and just aside here, you can see lots of people contributed to these studies, so I didn't really do it on my own with Inge. The press went nuts. So obviously, there's something special about the giant squid that makes it worth having a genome sequenced. We got mentioned in a website that where science meets science fiction. Not sure that is a compliment. There was like some references to monster DNA, and we also got mentioned in Forbes, although this has nothing to do with money. I did not know Forbes had a science column. So it was really fun. We got mentioned on Reddit, and just to say, there was a title called Unprecedented Gene Study Suggests Giant Squids May Be Massively Intelligent, reminding us as well, again, how important it is that we are conservative in what we say in our scientific articles. This comment actually got removed by Reddit because it was a biased headline. So they have high standards when it comes to the truth. Cephalopods. So giant squids is a cephalopod. It is a very, very diverse group, around 800 species. And there was actually no cephalopod genome sequence when we started this. Now we have like 5 or 6. out of 800, right? So still very few genomes available for this group. They are top predators with a very complex neuronal system compared to the other invertebrates, which makes them really interesting to study. And they've been around for around 600 million years, so they're a special group. Giant squids themselves are very, very mysterious. They live in the depths. As I said before, we can rarely see them. We do see their marks on the skins of sperm whales. They keep fighting sperm whales who actually eat them. So we often find beaks of giant squids inside the stomachs of sperm whales. And my colleague Inga used them for population analysis, actually. And so we know they're giant and they live in the depths of the ocean. And Inga, by studying their mitogenomes of 34 individuals, she did find that the mitochondrial diversity is really, really low. So she concluded that at least we got to know that there is a single species. Some other people had suggested up to 21 species. And it seemed to be like one big panmetic population. Of course, this could be a bias because of the type of data, so it could be a problem with the mitochondrial markers. So at the time, Tom got the idea that why don't we just do a genome and try to do some analysis with nuclear markers. And we actually wondered whether a genome would help answer some of all the other questions we have about giant squids, such as, you know, how quickly do they grow? How big do they really get? How fast can they swim? So could we, by inference, by comparative analysis, actually come up with some answers for these questions? I was very interested as a biochemist in the evolution of growth factors. Would that have an impact on the size that they can actually reach? And so we set up to do the genome. First challenge was getting a sample that was not too degraded so that we would have high quality DNA. And we did get it from some New Zealand collaborators. It's not the one in the picture, but just for you to see, it's kind of similar. So she was half that. But we did get a chunk of tissue that was enough to do some lab work. But it was a degraded sample. These samples have a elevated amount of ammonia because that's how they work out their buoyancy in the depths of the ocean. Lots of sugars, very, very sticky material. And so it sabotages the normal protocols that are usually developed for other types of species. Inga did a very good job in the lab, and so you will see the results of that. On my side, I do mostly the bioinformatics work. There was a predicted ancient genome duplication. We were afraid of that, and we managed to see that it was not the problem. The problem was the high heterozygosity that was really making it difficult to assemble the homologous chromosomes as one. So it's a recipe for disaster. It did become our badly behaved pet project. So it was a project I did on my side time, on a spare time, and also all the other collaborators. So kudos for that. We got some daily inspiration from some mugs. This is my lab mug right now. So Tom got us this mug that says, yes, you can sequence the genome of the giant squid. And of course, we know they can be sequenced, but the problem comes afterwards, right? We did keep on sequencing. We used all possible technologies. Of course, because of the curse of the giant squid, as we call it, none of them really worked at first. The first emails we got from all the teams was that we had no luck. There was difficulties in building the libraries. The DNA was not good enough. So it was a bit of a challenging project. We got some really bad assemblies using our short Illumina reads. The best assembly with a 32 KB and 50 was done by the Dovetail team using Miraculous. And looking at the K-bird distribution, we could see that the genome size should be around 2.7 gigabases, and in areas that do not overlap gene predictions, there were quite a lot of misassemblies. So homologous chromosomes being assembled probably into two, which caused them to be at half depth. The Dovetail data really made a big difference. So the Dovetail people were great, so they really helped out making this project a success. The N50 went up to almost 5 megabase, and then we had a slight improvement using the PacBio 10x depth PacBio data. The number of annotated protein genes, of course, is much lower in the improved assembly. It started off by being 100,000 in the Illumina assembly, so obviously a lot of false positives, a lot of gene fragments that were being annotated as separate genes. And so we had a very big improvement in our annotations. 33,000 protein-coding gene annotations actually have transcript evidence. I did sequence the transcriptomes of several tissues from 3 other Oegopsis squids. Obviously, for the giant squid, it's impossible to obtain transcriptome data because the tissues are quite degraded. But using these other squids, I was able to see that for 30,000 annotations, around 30,000 annotations, we did have evidence that these genes were real, and 3,000 of these genes, so around 10%, were actually cephalopod specific, with no orthology inference for other species and no way to know what is their function. So perhaps holding a lot of information regarding cephalopod specific features. The genome contained almost half of repeats. Almost half of the genome was made of repeats. The genome of the giant squid was not a very giant genome, so it was comparable to the octopus V. maculoides. So there's the other cephalopod genomes out there are much bigger, almost double. But the content of repeat types was pretty similar to other cephs. So we could see that all the Hox genes are found in the same scaffold, albeit with really large intergenic distances. So for the previously published squid and octopus genome, they were fragmented, probably because of assembly issues. And what we could also see is that between the Hox genes, we seem to have some open reading frames. So potentially, and that is very, very unusual, there could be other genes being translated between the Hox genes, which might or not have an impact on their complex body plan. And I just want to finish by saying that this was, again, a very big team effort. I keep adding collaborators to this project. It was a lot of fun to meet all these different people and to meet the cephalopod people in specifically. And thank you. You're muted, John.
Oops, I forgot to unmute. Yeah, sorry. Thank you for the great talk. I was wondering, what was your DNA size fragment from your preps? How fragmented was the DNA in the final DNA extraction used for sequencing?
There was a big blur. The biggest fragments we got were around 40 kb. And then there was like a big spot.
Yeah. And did you think that the mucus of your organism was still posing a problem at that stage, or do you consider that you resolved the mucus problem completely with your final DNA extraction preparation?
Well, this was Inga's work, so I myself was not part of the optimization of the protocol, but that was exactly what she did. The initial protocols she was using were not appropriate for polysaccharide-rich tissues, and she did eventually, after 6 months trying, was able to get rid of that problem specifically. And so the fragment size, of course, the biggest ones were around 40 kb, but I mean, most of them were in the 2 kb range and smaller, right? So, and that was a problem with the sample quality.
Thank you. Wondering, so how did you annotate the genome precisely?
Which program was used to annotate the genome? It was a program developed by people in Bratislava that uses, that is specific for organisms which do not have close relatives in the database as well annotated. I can't remember the name right now, but yes.
Is there any specific reason why you didn't choose one of the commonly used, like Maker or Breaker?
Yes, because precisely if this program was developed to deal with the deep divergence between the genomes that have been already annotated. So it uses some de novo annotation, but it also uses input from already annotated genomes. So it allows us to have less false positives.
Thanks. One remark from Rob Waterhouse that sometimes finding genes between the Hox genes is actually totally normal, that people omit it from the figures in papers, but it's not something to be surprised about.
Oh, okay.
That's a good point.
But we were surprised to find those genes and the large intergenic distances, right? So perhaps it's actually less less surprising that here with the large intergenic distances, there are ORFs that might be translated, right?
And maybe one final question, very open. What are the next steps for this project?
Well, actually, we performed a comparative analysis that has not been published fully together with the genome paper. And it will be integrated with a number of transcriptomes from other cephalopods, given that it's impossible to actually do it for the giant squid with different tissues, and try to understand whether those 3,000 genes that are specific to cephalopods and for which we have no annotated function are specific to different tissues in the cephalopod range tissues we have.
Thank you very much. Our next participant will be Gabriel Gonzalez Sonoda.
So, hello everyone, I'm Gabriel, I'm from Universidade de São Paulo, and today I'll present my talk entitled Evidence for Duplications and Positive Selection on Toxin Genes in Nemertia, a Nomic Approach. So, first of all, let me remember you who are the Nemertia. So, Nemertia is a phylum composed of marine animals and mostly bentonic too. There are around 1,300 species, and most of these animals produce toxins which they use for both predation and defense. This picture shows a bit of the diversity of the phylum. So the synapomorphy of the group is the presence of a proboscis, which they use for both predation and defense, but it spends most of the time inside the animal in a cavity called wrinkle cell. So here we have on the left an animal averting its proboscis, and on the right the same thing. We have this red animal averting a beautiful white branched proboscis. Nemertia belongs to Spiralia in the metazoan phylogeny, according to the most recent publications on metazoan phylogeny. And according to Laumer and colleagues, more specifically, it's the sister taxa to Annelida. This phylogeny of Nemertia shows the main groups of Nemertia and their relationships. So we have Palanemertia as sister taxa to the remaining Nemertia, and Oplonemertia as a sister taxa of Pylidiophora, and Pylidiophora containing Heteronemertia and Obrechtidae. Now talking about toxins. Toxins have evolved many times in the metazoan phylogeny, but even though there are many groups which produce toxins, just few of them have been receiving proper attention in regard to the evolution of these toxins. And these are the spiders, scorpions, hymenopterans, cone snails, and snakes. While the remaining groups remain kind of neglected, such as the Nemertia that don't even appear in this review. So what is known so far about the evolution of toxins? Well, one hypothesis to explain where they come from is that they arise from the duplication of a physiological gene. Once the physiological gene has gone under duplication, one of the copies may be expressed in the venom gland, where it will go under selective pressure independent of its counterpart expressed in the body tissue. After that, so it will eventually become a specialized toxin gene. And after that, more duplications may happen, giving rise to more copy of these genes, which then may go under new and subfunctionalization, leading to a greater diversification of the venom. Another important idea to understand the evolution of toxins is the red queen hypothesis coined by Van Valen, which states that you must run to stay where you are. So how this would apply to toxins? We would have that the toxins are applying toxins to, the toxins are applying a selective pressure to the target that would develop a resistance. And so this resistance would reciprocally exercise a selective pressure to the toxins. So there would be a positive feedback for both the toxin and the resistance to keep changing all the time. From a molecular point of view, this could be translated into positive selection. It is also proposed that such a regimen would result in the fixation of genetic copies, since this would increase the arsenal of the producer of the toxins and making it easier to overcome the resistance of the target. And what is known about evolution of peptidic toxins or the diversity of peptidic toxins in Nemertia? Unfortunately, few peptidic toxins have been isolated from this group, but works such as this one published by Whelan and colleagues show that but works such as this one, published by Whelan and colleagues, have searched the transcriptome of many emerging species for putative toxin transcripts, and they have found that there might be a great potential of putative toxin transcripts in this phylum. So the main goals of this work are to identify putative toxin genes in the transcriptome of 3 different emerging species, and to compare the evolution of toxins in emergence to that of other animals, and that is to find evidence for positive selection and duplications. So first of all, which animals we collected for that? We have sampled Linus sanguineus, which is an heteronemertian, and we also have sampled the Oplonemertians, Nemertopsis bivitata, and Autochflonemerts eneba. It is worth noting that these are the first transcriptomes of this genus. So once we had the transcriptomes, we searched for the putative toxin transcripts using trinotate. So we searched for the gene ontology term toxin activity, and we also searched for peptide signals in the products of these transcripts that would indicate that these toxins go to the secretory pathway, as it is expected for the toxins to be secreted. So once we had the putative toxins, we got the orthogroups using OrthoFinder and some transcriptomes downloaded from SRA as our transcriptomes. So we obtained the orthogroups that contain these putative toxins, and we have codon aligned these orthogroups, which made it possible for us to do selection tests using CodeML and the site model, and also made it possible for us to do duplication analysis using DSI with phylogenomic tree as reference. We also have done duplication and expansion and contraction analysis to the phylogeny of the Nemertia. So to do the duplication analysis, we used the OrthoFinder duplication inference algorithm with orthogroups which had at least 80% occupancy plus a phylogenomic tree as reference. This yielded us with a list of duplication events. For the expansion and contraction rate, we used CAFE to estimate that with a non-traumatic tree. And so we have obtained the rate of expansion and contraction per gene family per time, which is lambda. Now the results for the Autochflonemerts eneba, we have identified 17 transcripts of putative toxins corresponding to 7 different genes. For Nemertopsis bivitata, 6 putative toxin transcripts corresponding to 4 different genes, and for Linus sanguineus, 44 putative toxin transcripts identified as 23 different genes. Now, this is interesting because Linus sanguineus are heteronemertians, which are the only group which had peptidic toxins isolated from so far. And so this indicates that these 44 putative transcripts are not false positives, but are in fact toxin transcripts. So once we had the putative toxins, we proceed by doing an evolutive analysis of these toxin genes. And so far we have only done such analysis on 4 genes for a matter of time. So we searched for positive selection and duplication events, as I've told before. And these are the results. We found evidence for positive selection in half of these genes, and on the other half, there was evidence for purifying selection. So how can we explain such purifying selection in toxin genes? Well, we have hypothesized 2 scenarios for that, and they are not mutually excluded. So the first scenario is that there are a wide diversity of targets, and the second one is that there is a low impact on the target population. So both of these scenarios would lead to the absence of a selective pressure for the target to develop a resistance. And if the target don't develop a resistance, it will put an end to the positive feedback of the red queen hypothesis. So there would be no positive selection, just purifying. Now about the duplication events, we have found duplication events in all of these toxin genes, but what's most surprising is that all of these duplications were assigned to nodes belonging to Heteronemertia, which again is so far the only Nemertia group which has proteinaceous toxins isolated from. And this make we ask ourselves, did Heteronemertia experience more gene duplications than the other Nemertia? So to investigate that, we used AutoFinder to count the duplication events in each node. So in this graph is the number of duplication by node. Each different bar is a different node. And what we did was to plot all these nodes with a high number of duplications in the phylogeny of Nemertia. And what we found out is that all of them belonged to Heteronemertia, indicating that Heteronemertia had, in fact, more duplications in that data set of genes with at least 80% occupancy. Next, we estimated the contraction and expansion rate, that is lambda, in these gene families. So it's a measure of how many contractions and expansions, that mean deletions and duplications, happen in given amount of time in given family. So first we estimated a global value for the whole phylogeny, and we have obtained this value of lambda. And next we estimated two different values, one for the Heteronemertia and the other for the remaining Nemertia. And what we found out is that the lambda value for the remaining Nemertia is very similar to the global value, while the lambda value for the Heteronemertia is slightly higher, indicating that the gene families in Heteronemertia expand / contract kind of faster. But since we are not interested in the contraction, we are interested in the expansion to explore the evolution of toxins. We've done a third estimate of only the expansion rate per gene family per time, doing the same thing, one value for the Heteronemertia and the other value for the remaining Nemertia. And once again, we have found a higher value in Heteronemertia than we have found in the remaining groups. So with these results, we have found more gene duplications in Heteronemertia, and we have found that the gene families in Heteronemertia are evolving faster and with a higher expansion rate. We have obtained these results with two different softwares, with Coffin and AutoFinder. And once again, Heteronemertia is the only group which had peptidic toxins isolated from. And as we have discussed before, duplications are an important process in the evolution of toxins. So this leaves us with a final question, that is, if these duplications have anyhow made it easier for the evolution of peptidic toxins in the Heteronemertian group. And that's the question I will end my presentation with. So I'd like to thank all my research group. I'd like to thank John Arenberg for teaching me a lot about Nemertia. I'd like to thank Sebimar, which aided us in the collects, and thank CNPq and FAPESP for the funding. Obrigado.
Thank you very much, Gabriel. Very nice.
Hey there.
Thanks. So there's a bunch of questions we'd like to ask you. First question was, what was the completeness BOOSCO scores and what database did you use?
And so for the new transcriptomes that we assembled here, we used the Metazoan database since it's the closest database that we have for Nemertia. And as a completeness for these three new transcriptomes, we had more than 90% for lineosanguineous and no less than 85 or 80, I guess it was 83% for the both Hoplonemertians. So it was, they were kind of complete for this database, at least.
Thanks. Another question is about toxins that can often be rather short peptides. So did you have to use some specific approach to correctly find or predict orthologs and homologs?
So unfortunately, we didn't. We actually searched for peptides that had at least I think it was 50, which is the default size for transdecoder. But we still got to find many putative toxin genes. It's no forwarding that by this approach, we didn't recover a new transcript of toxin that has been reported in 2018 that is alpha-nemertide. We couldn't find it by this approach, but still we got manipulative toxin genes.
Thank you. I'm also wondering about the duplication that you show. So of course you have only transcriptional data, but do you try to look in genome data to see if it's more like tandem duplications or very remote places where the new copy lands?
Well, that's a very nice question, but there is actually just one genome for Nemertia, and it's not actually very, it's not annotated, it's not complete, so there is not such a data that we could use.
Okay, and there is one question: how do you find which transcripts belong to the same gene?
So we use two approaches in this work. So for the CAF approach, we use a reciprocal blast and ortho and an MCL clustering to identify transcripts that were closer to each other, and this would be the same gene. And for identifying the toxins and for identifying duplications, we use OrthoFinder, which uses a similar approach, but it uses bow tie and a different clustering algorithm.
Thank you very much. I think now it's time to introduce our next speaker, who happens to be a PhD student in my group, Nadège Guglielmoni.
Hello, everybody. My name is Nadège Guglielmoni, and I am a PhD student at the Université Libre de Bruxelles and a member of IGNITE. My topic today is how to crack the genomes of non-model invertebrates, lessons from coral and rotifer genome projects. Here you have a survey of assemblies of non-vertebrate animals with the scaffold N50 and sequencing methods used. You can see that there were already some very good assemblies of non-vertebrates with the first sequencing methods. And with the advent of short-read sequencing, genome assemblies became more available, and but with a low contiguity. Now with long-read sequencing, often combined with short reads, there are even more genomes available for more species of different phyla. Of course, these high N50s are not only due to long reads, but also to efficient scaffolding methods. So here you have an example with assemblies that included a high-C scaffolding step highlighted in blue. And except for some outliers, you can see that they form a cluster of assemblies with scaffold N50 over 1 megabase. High-C is becoming more accessible now in terms of protocol and appears as an appealing technique to obtain chromosome-level assemblies for challenging species. That is why for recent assembly projects, we have decided to use a combination of short reads, long reads, and High-C. Today, I'm going to present to you the results of two genome projects we finished recently. For the coral Astrangia puculata, we originally had an assembly that was only half the expected size. And for the rotifer Adenita vaga, the first assemblies we tried were quite larger than the expected size, up to 160%. We have been working on the genome of Astrangia puculata with two collaborators, Liliana Bohms and Catherine Stankiewicz, and they had a haploid genome size estimation of 453 megabases, while the first assembly with a combination of short reads, Chicago, and High-C was 252 megabases. As they already had a high coverage Illumina and High-C, we sequenced low coverage nanopore reads that we assembled with WTDBG2, then polished with short reads using Hypo, and scaffolded with High-C data using Instagrail. With this strategy, we obtained 455 megabases assembly in 14 scaffolds with a Busco completeness of 90.4%. Here you have the high-C contact map of Astrangia populata after scaffolding, and you can see the high-C interactions of the whole genome versus the whole genome. This portion of the genome has high interaction with itself and much less interactions with the rest of the genome, as this is one chromosome, and all the different squares of high interactions correspond to one chromosome. So you can see the 14 chromosome level scaffolds. This new assembly brings incredible improvements compared to the old assembly, as it is much less fragmented and much more complete. And this shows that low coverage long reads can resolve regions that were not solved by high coverage Illumina. For the genome of Adinetavaga, we had a much different problem. The expected haploid size was 102 megabases, and we had both PacBio and nanopore reads. But when we tried to assemble one or the other dataset with 7 different assemblers, all the assemblies were much larger than the expected size. Why is that? The genome of Adinetavaga has variable levels of heterozygosity with regions of high heterozygosity. Here you have an example of a diploid genome with 2 haplotypes and homozygous regions and a region of high heterozygosity. During the assembly process, the assembler needs to collapse the 2 haplotypes to produce a good haploid assembly, which is easy for homozygous regions, as they are identical, but much more difficult for highly heterozygous regions. To get a good haploid assembly, the assembler needs to choose between the 2 versions of the region, but they often fail to do so and output the 2 versions of the same region, which leads to a duplicated region in the assembly. To obtain better collapsed haploid assemblies, we defined 3 strategies. The first strategy is to choose a better assembler, and for that we tested 7 long-read assemblers to see which one were better at collapsing haplotypes. The second strategy is to use only the longest reads for assembly, and we also tried to remove uncollapsed haplotypes from assemblies of all reads using the purging tools like haplomerger 2, purge depths, and purge haplotics. We compared the results on 2 long reads datasets of PacBio and nanopore reads. So we looked at the coverage distribution after mapping the long reads to the contigs. And you can see that for all assemblies, there is a high coverage peak for properly collapsed haplotypes. And for most assemblies, there is a second peak at about half the coverage of the high coverage peak. These are the uncollapsed haplotypes because half the reads map to one haplotype and half to the other haplotype. You can see already that the Ra and WT-DBG assemblies have almost no half coverage peak, which indicates that they are better at collapsing haplotypes. You can also see that for some assemblers, there are low coverage contigs that are contaminants and can also contribute to the increased assembly size. For today, we'll look at the results of PacBio assemblies. And we compared the results of assemblies with all reads, assemblies with only a subset of the longest reads, assemblies of all reads after purging with haplomerger 2 or purge depths or purge haplotics. For assemblies of the longest reads, the next de novo and Ra assemblies are close to the expected size. For haplomerger 2, all the assemblies are reduced, but except for the canoe and maybe the raven assemblies, the assemblies are shorter than we would expect. With perch depth, the flea and next de novo assemblies are close to the expected size, and with perch hapletics, the Shasta and WT-DBG2 assemblies are close to the expected size. Now for the coverage distribution, we see that for assemblies of the longer streets, the assemblies produced by next de novo and raven and Shasta have a smaller half coverage peak. And the low coverage contigs are absent or almost for the assemblies of raven, Shasta, and WT-DBG2. With haplomerger 2, the half coverage peak is reduced for the assemblies of canoe, flea, next de novo, raven, and Shasta. And for the assemblies after perch depth, there is also a reduction of the half coverage peak for canoe and then flea, next de novo, raven, and Shasta. After perch hapletics, the assemblies of canoe, next de novo, and Shasta have a reduced half coverage peak, and the low coverage contigs are removed. What we learned from this project is that some assemblers are better at collapsing haplotypes, Ra and WT-DBG2, that assembling only the longest reads can improve structure, and that hapletics purging tools work better combined with specific assemblers. We also tried to combine two purging tools, perch depths and perch hapletics, and obtain better results for canoe and raven assemblies. Here is an example of high-C scaffolding with Instagrail for an assembly produced with only the longest reads and Ra. And we obtain 6 chromosome level scaffolds as expected. So we have seen that long reads can efficiently build structurally correct haploid assemblies, that high-C scaffolding is also efficient to obtain chromosome-level scaffolds for non-model invertebrates. And these strategies are also applied to other genome projects in IGNITE, such as the mollusk Arion vulgaris with 26 chromosome-level scaffolds. And I've also been working on the Catognas Flaccissagita and Flata, for which we recently obtained 9 chromosome-level scaffolds. These are all the people that contributed to this work that I would like to thank. And thank you for your attention. I will now take your questions, and you can find these slides on my GitHub repository.
Nadej, it's nice to see all this progress in non-model genome sequencing. Here are a few questions from the public. The first question is, how many genomes did you use for your survey? Was there any special requirement for a genome to be included in the survey?
I suppose it was for the first survey of the different assemblies of non-vertebrates.
Yes.
So the requirements were for the assemblies to be from non-vertebrate animal species. We tried to look through all the publications we could find. But I think, although for the arthropods, we have not been completely thorough, as there are many insects genomes available, so they are quite overrepresented compared to the other phyla. I think we're up to 265, 62 genomes right now.
Thank you. Another question about the rotifer. Where did the expected genome size come from? Did you do flow cytometry or something similar?
So for this paper, we've been using the genome size estimation that has been calculated using Kmer spectrum with CAT on the Illumina data. But for the genome paper, they've also did a genome size estimation with flow cytometry, but it was solving tuned at the time.
Another question, if collapsing the haplotypes was so difficult for this species, then wouldn't it be better to just separate the haplotypes and to get a diploid assembly?
Yeah, I really would like to separate the haplotypes. Yeah, that's a good question. Yeah, so collapsing haplotypes is challenging, but separating them is also challenging because then you have to combine the right alleles from one heterozygous region to another. So for different, it's difficult because then if you want to do that, you need to have all your sequencing data from one single individual. But that's also what we are now trying to do with the genome of Adineta vaga, and we've also been working recently on a tool for phasing assemblies using Hi-C for phasing assembly graphs with Hi-C.
Thanks. Yet another one. How did you identify the low coverage contigs as contaminants, and why are they recovered in some assemblers and not by some other assemblers?
So the low coverage contaminants, low coverage contigs, we identify them as contaminants with blob tools, and I think, I don't know, but I think that some assemblers just automatically remove low coverage contigs from the output, I think.
And a final question. Someone was wondering if you encountered specific problems to do IC for some of the species you just showed.
For the Hi-C protocol, so for the coral, we were not the ones to generate the Hi-C data, but I've prepared a Hi-C library for the rotifer Adineta vaga. And for this one, I must say that it was all right. We used a Arima Hi-C kit and it worked fine. I've also been working on this protocol on other species, so on Ketogonas, for which it worked quite well, and also on sponges. There it was a bit more difficult, and we had to adapt the beginning of the protocol, but then worked also nicely.
Thanks a lot, Nadej. And now we're going to move on to our last speaker, Nitsala Karunu-Daisiri.
Okay, well, good afternoon, everyone. I'm honored to share with you our experience on deciphering genome of the black tiger Chimpanzees monodon, and hopefully that can shed some light on crustacean genome evolution. So the motivation of this study is basically the growing world population. You can see that last year, our world population actually reached over 7 billion, that's 7.7 billion. And by 2050, that will reach 9 billion. The big question is, how are we going to feed all these people? Fishery product actually has been a major resource of nutrient for humankind, mostly protein. In the previous times, aquaculture and capture production actually had contributed equally to the fishery production, but since 1970s, aquaculture actually had surpasses their capture production by far. And why is that? That's due to perhaps the limitation of natural resource, as well as how we try to not overexploit the natural resource. So the key to providing enough food to the growing world population is to make sure that aquaculture is sustainable and is effective. Among edible crustaceans, pinnate marine shrimps are the predominantly cultured groups, and that actually contribute to over 4.5 million tons per year on average. Black tiger shrimps or Penaeus monodon actually account for 9% of total crustacean production. So you can see the significance of this species. you see the significance, and you may ask why up to now the genome has not been sequenced, because of lots of challenges that we face for this genome. It had been reported that it's really difficult to isolate high molecular weight DNA of this species, because there's a DNA activities for this species. Also, the high percentage of repetitive sequences have been reported that it could be up to 50%, but you can see from our report later that we actually even higher percentage of repetitive sequence in this genome. The shrimp itself has really large genome. It was estimated to be about 2.6 gigabyte, which is GB, which is about 80% of human genome. And it had large number of chromosomes of 2n equal to 88. And also up to now, there's lack of high quality physical map. But we finally get it done. And how do we do that? We actually use the traditional approach of sequencing technology, but we use the advanced technology. And I'm going to go through that really quickly. For de novo assembly, we employ long read sequencing platform of PacBio, and we were able to get 77x coverage from PacBio on the average length of over 10,000 base pair. And you know that this technology sometimes have some error, so we actually do error collection using Illumina short read sequencing platform, and we get 133x coverage from Illumina short read. But to scaffold them together was a challenging task because of the repeat embedded in the genome. So we actually employed the dovetail genomics based on proximity ligation approach. And with that, we were able to obtain the chromosome scaled genome assembly. And you can see this little blue box here. We get 44 boxes here. That's actually equivalent to the number of chromosomes. So if you can see assembly statistic here, that you can see that we get 44 contig, which is equal to the 44 chromosome. And also, after we get the scaffolded chromosome, what we do is we have to do gene prediction. So we use a computational evidence based approach supported by our transcriptome data. So recently we published a report on a novel full-length transcriptome data in Frontier in Marine Science, and you can see the details of our data there. But basically what we did was we used a 10 major tissue of this shrimp, and we sequenced full length, and we can obtain tissue-specific transcripts or the common transcript. And with that, we use it to help predict genes and support that our gene prediction is correct. And what we get is that we predicted to have 31,000 gene models, and out of that, 65% can be annotated. And out of that, actually, we have 85% that have evidence from our transcriptome data. So that's pretty good. That's saying that our assembly, the scaffolded genome probably is correct and with high quality one. Then we did some annotation. And I will conclude quickly that the assembly P monodon chromosome actually contain 44 chromosomes, totaling up to 2.39 gigabase assembly. In terms of repeat sequences, as other people were reporting to estimate of 50% repeat in the chromosome of shrimp, for the black tiger shrimp, 2.5%. That's a huge amount of repeats in chromosome. And for gene density, we get 672 genes per chromosome. That is normal, so people can get hundreds to thousands of genes per chromosome. GC content, our GC content is 36%. That's about the same as the white shrimp, its cousin shrimp species. So that's pretty good. And from the available chromosome that we obtain, we can then do some genomic comparison. So first, we actually compare with its cousin of L1-amide. So from their relationship, you can see here, most of the gene has 1-to-1 relationship. That means you have one copy on one chromosome, and you of the P monodon, and that have one copy in the chromosome of L1-amide. With this high degree of conserved synteny, suggests that P monodon and L1-amide probably derived from the common. However, we found a few pair exhibiting 1-to-2 relationship between the P monodon and L1-amide. That's, for example, you have genes in chromosome 4 of P monodon, but have 2 autologous genes in chromosome 14 and 15 of L1-amide. Similarly, you have genes from P30 or chromosome 30 in P monodon, and presented as two genes in L21, L22. This kind of relationship probably suggests that a certain chromosome derived from the common ancestor of P monodon and L1-amide were fragmented into 2 smaller chromosomes in L1-amide, but remain as a single copy in P monodon. Alternatively, it could be that the assembly or scaffold of L1-amide chromosome might not be correct. It could be actually 1 chromosome together, but they were scaffolded into 2 chromosomes. So that is how we actually see this data. We also construct phylogenetic trees of the mitochondrial protein coding genes of these arthropoda organisms, and you can see that, as expected, organisms from the same order of Decapoda, including P monodon and L1-amide, actually group together. And you can see how closely related between the Pacific white shrimp of L1-amide and P monodon. They pretty much share very close relationship. Interestingly, though, their organisms from their order of Decapoda from class of Maracostraca is more closely related to class Insecta than other organisms from the same class. This is just for mitochondrial protein coding gene phylogenetic tree. Now, if you look into nucleus genes, we can also do comparative genomics of relevant species. We use 5 crustacean species here, and we use 1 tuna fish as an outlier. And this is actually might be a little bit busy graph, but I'm going to walk you through it. This is upset pot here. Upset pot is basically just simplified version of Venn diagram. But you will see that we have 6 species, and if you have 6 circles of Venn diagram, that could be confusing as well. So the way to read this is this X, the bar graph here, it actually represents the number of protein family, and the dot here, right here, for example, if the dot here presented in E affinis here, that means this number of protein actually presented only in this species. If you have 2 dots here, you can see this 5,000 gene presented, 5,000 protein family presented in P monodon and L1-amide. With this together, you can see which one is closely related, which one is actually being shared between species. So if you look into phylogenetic trees here, you can see that L1-amide, P monodon, and preferred H. estaca here actually share close relationship here. That's actually kind of, to me, surprising, because L1-amide and P monodon actually marine organism, whereas H. estaca is actually fresh water crustacean. And also H. estaca actually has much smaller genome. The shrimps have about 2.6 GB, whereas this freshwater crustacean has only 1 GB. But they seem to share common recent ancestor. So what could have played a role in the expansion of genome size, we actually when hypothesize that it might actually be the fact that TRIMs expand the genome size by acquiring other genetics elements such as lipids. So we look into the lipid analysis of this organism and found that, oh well, actually this graph, this on the left hand side actually is the absolute number of lipids. The right hand side is actually percentage of lipid or relative number of lipids based on the genome size. So you can see this with this organism here, P. monodon has the highest lipids and which is very similar to L-vanamide. But you can see here, H. estaca, the organism that had shared the common ancestor with L-vanamide and P. monodon, actually has much less lipid. So yeah, maybe lipid actually explain the expansion of the genome size of the pineal TRIMs. On top of that, you can see the contributions of the distribution of the lipids, and with all these examined organisms, you can see groups and yellow box here play a contribution toward their L-vanamide and P. monodon, and these two boxes belong to low complexity and their simple lipid. So we think these two type of lipid actually explain the large portion of the genome size variation in decapoda organism. So in summary, we have achieved the chromosome level genome assembly of n equal to 44. We see 1-to-1 and 1-to-2 synteny relationship between black tiger shrimp and Pacific white shrimps, and from mitochondrial genome comparisons, we see that black and white shrimps are closely related. The order of the decapoda seem to be more closely related to class of Insecta. From nuclear protein phylogenes, we found also that black and white shrimp as well as H. estaca are in the same clade, and perhaps repeat element might contribute to the genome size and function. And hopefully together that is the start of the new era where genomes of the shrimp is available, and we can speed up the knowledge of shrimp biology to contribute toward our food security. So I thank you for your attention. Thank you very much.
Thanks a lot, Nisa, for this inspiring talk. I got a bunch of questions to ask you. So one first question is, why do you think the chromosome count is so high in this species? And are you able to estimate the rate of recombination? And if so, do you see the expected about one recombination even per chromosome?
Well, I have no idea why the number of chromosomes in this species is really high, but this is not surprising because if we look into the white Pacific white shrimp, the number of chromosomes is as high. So it's not something that strange with the shrimps, but it could be the fact that the shrimp itself had been evolved over time. I'm not sure, I'm just venture the guess right here, giving that there's different adaptation, you know, over the evolutions. So then maybe that's explain number of chromosome. I just not quite sure. Yeah.
Because also you showed some event when one chromosome was giving two very unequal size chromosomes, one tiny one and a large one, according to the genomic data.
Yes.
Did you see any correlation of that with the karyotype? Do you see tiny chromosomes in the karyotype, like big ones and tiny ones?
Yeah, we didn't learn the techniques ourselves. So, but we're not sure if that correlated to the techniques, the result of that techniques. Yeah.
Because it would be very interesting to do fish, for instance, to verify it on the chromosome.
Definitely. We're going to explore to that because we didn't even, to be honest, we didn't even expect that we're going to get number of contigs equal to the number of chromosome, because this species has been very difficult and really challenging. People have been trying to doing genomic sequencing for years, for decades, I would have to say, and no one had succeed in this. So when we got the number of contig equal to number of chromosome, we think that, okay, well, we gotta have to explore this further, because we're not the evolutionist people. We just want to use the sequence data to do some other studies, such as reproductive maturation studies and immunity system and so forth.
I think we have time for one more question. Why are the branch lengths on the 6 crustacean tree you showed? Why is that so different?
Which slide is that?
I think this is a question asking you, like when you have the one showing the shared gene between pairs of taxa, on the upper right, you had a tree with very different branch lengths for the crustacean taxons.
Well, basically, it's, if you see some of the, I'm not sure if you're talking about the slide number. Which slide do you know?
I think it was towards the end of the presentation. It was a question asked like 3 minutes ago. So it was towards the end of the presentation when you were showing the gene set and the genes that are shared between different taxa.
Why is it different?
Why you have such a very large difference in branch length?
Because what happened is we do 2 comparative genomics. One is mitochondria. We're using 14 genes to do that. One is the nucleus genomics, and we look into the upset part. So if you're talking about the mitochondria, it could be that the 14 gene that we selected are not the optimal or set of genes, or the branches are not what we expected. Is that the question?
I think the question was more about the upset graph, because there you have mixed species. Yeah. But I mean, I think the time is already there to close the session. So I would like to invite all the speakers to come together again, to turn on the camera and to finally close the session. So thanks everyone for great talks, great question and answers, and for being there. And for the final words, I'd like to pass the microphone to Joe, President of GIGA, for officially closing the session.
Thanks, Jean-Francois. And thanks for all our speakers who came today. It's a very good session, great session to have you here. Shows the diversity of our group, fascinating animals, and so much more to come in the future. So I know we're going into the concert time, but we're still planning on a meeting. We hope we could have one next year, and with the success of this type of meeting, online or maybe hybrid online, but most likely the way things are going, maybe just online, it still might be good enough. So stay tuned. Jean-Francois is our conference director, so I think hopefully we'll send some news out in the next few months about some plans for a GIGA meeting. And now we got our hands full with maybe some good applications too for the scholarship.
Thanks, Joe. Thanks, everyone. And yeah, stay tuned. Bye.
Bye. Thanks, everyone.